Good morning, everyone. Today, we're going to dive into one of the most fundamental processes in all of biology, the cell cycle. But we're not just going to talk about the phases G1, S, G2, and M, which I know you've all encountered before. Instead, we're going to get to the very heart of the matter, how this incredibly complex process is regulated. Why does it matter so much? Because a single misstep, a single failure of regulation, can have catastrophic consequences, the most well-known being cancer. Think of a cell as a sophisticated, high-stakes factory. The cell cycle is the assembly line, and the product is a brand new, genetically identical cell. What would happen if this assembly line just ran nonstop without any checks or balances? The product would be defective. In the case of a cell, a defective product could mean a cell with damaged DNA or with the wrong number of chromosomes. A functioning factory needs quality control at every step. That's precisely what cell cycle regulation is. It ensures the cell is properly prepared before it enters a new phase, that it's checked for DNA damage, and that it has all its components lined up perfectly before division. Without this rigorous oversight, we would see a massive increase in mutations and cell death. So, let's begin by looking at the key players that orchestrate this complex dance. At the heart of cell cycle control are two groups of proteins that work together as a team, Cyclins and Cyclin-Dependent Kinases, or CDKs. This is probably the most important concept we'll cover today, so let's make sure we're all on the same page. First, let's talk about the CDKs. As their name suggests, they are a type of kinase. A kinase is an enzyme that adds a phosphate group to another protein, a process called phosphorylation. This action can either activate or inactivate the target protein, essentially acting as a molecular switch. But here's the key: a CDK is inactive on its own. It's an engine without a key. It's ready to go, but it can't function. That's where the cyclins come in. Cyclins are the other half of this partnership. They are a family of proteins whose concentration cyclically rises and falls throughout the cell cycle. Unlike the CDKs, they are not enzymes. Instead, they are regulatory proteins. They are the key to the CDK engine. A CDK can't do its job of phosphorylation until a cyclin binds to it. It's the binding of the cyclin that makes the CDK functional. The combination of a cyclin and a CDK is called a cyclin-CDK complex, and this complex is what actually drives the cell from one phase to the next. Let's visualize this interaction a bit more closely. A CDK protein, on its own, has a blocked or non-functional active site. Think of it like a car engine where the ignition is locked. When a cyclin binds to the CDK, it causes a profound change in the CDK's three-dimensional structure. This conformational change is like inserting and turning a key. It unlocks the CDK's active site, making it capable of phosphorylating its target proteins. So, the functional unit that we need to remember isn't just the CDK, and it's not just the cyclin. It is the specific cyclin-CDK complex. Each unique complex is responsible for phosphorylating a very specific set of proteins, and it's the phosphorylation of these targets that allows the cell to transition from one phase to the next. For example, a particular cyclin-CDK complex might phosphorylate proteins that are required to initiate DNA replication. Another one might phosphorylate proteins that break down the nuclear envelope during mitosis. The cell is essentially a finely tuned machine, with different cyclin-CDK complexes acting as the switches that flip the cell from one gear to the next. As we just discussed, the cell cycle is driven by specific cyclin-CDK complexes. As you can see in this diagram, the concentration of different cyclins rises and falls at very distinct times. This cyclical pattern is what gives them their name and what allows the cell to progress in a sequential manner. Let's break down the main families. First, we have the G1 cyclins, primarily cyclin D, which bind to their corresponding CDKs to drive the cell through the G1 phase. Their job is to prepare the cell for DNA replication. As the cell gets closer to the S phase, the concentration of G1/S cyclins, such as cyclin E, increases. This cyclin E-CDK complex is crucial for pushing the cell past the critical G1 checkpoint. Once the cell is in the S phase, S cyclins, like cyclin A, take over. The cyclin A CDK complex is responsible for initiating and maintaining the process of DNA replication, ensuring it's completed accurately. Finally, as the cell prepares for mitosis, we see a rise in M cyclins like cyclin B. The cyclin B CDK complex drives the cell into mitosis and orchestrates events like chromosome condensation and the breakdown of the nuclear envelope. Notice how the concentration of each cyclin peaks and then quickly drops off. This rapid degradation is a critical aspect of regulation that we'll come back to, as it ensures the cell cycle is a one-way street. Now that we understand the engines of the cell cycle, the cyclin CDK complexes, let's talk about the safety features, the checkpoints. If the cyclin CDK complexes are the molecular gears that drive the cell forward, the checkpoints are the brakes. They are critical control points where the cell pauses to check for potential problems before moving on to the next phase. Think of them as the quality control inspectors on our cell factory floor. There are three major checkpoints we need to focus on, and they occur at key transition points in the cell cycle. First, we have the G1 checkpoint, also known as the restriction point. This is arguably the most important checkpoint because it's where the cell makes the fundamental go or no-go decision. Next is the G2 checkpoint, which ensures that all the cell's DNA has been fully and correctly replicated before it commits to mitosis. And finally, there's the M checkpoint or spindle assembly checkpoint, which is located in the middle of mitosis. Its job is to ensure that all the chromosomes are perfectly aligned and ready for segregation. These checkpoints are not just theoretical concepts. They are an absolute necessity for preventing errors that could lead to genetic instability and disease. Let's spend a bit more time on the G1 checkpoint because it's the point of no return. Once a cell passes this checkpoint, it is committed to completing the rest of the cycle. This is the stage where the cell assesses its internal and external environment. Is the cell big enough? Are there enough nutrients? Is there a green light from growth factors? Most importantly, is the cell's DNA damaged? Two of the most famous players here are the tumor suppressor proteins, P53, and the retinoblastoma protein, or RB. Let's start with P53. It's often called the guardian of the genome. When DNA damage is detected, P53 levels rise dramatically. A high concentration of P53 can trigger several responses. It can halt the cell cycle to allow for DNA repair. Or, if the damage is too severe, P53 can initiate apoptosis, or programmed cell death, to prevent a damaged cell from proliferating. Then we have the RB protein. Under normal conditions, without growth factor signals, RB acts like a molecular clamp. It binds to a transcription factor called E2F, essentially locking it down. E2F is needed to turn on genes required for the S phase. When the cell gets a green light from growth factors and the right cyclin CDK complexes, those complexes phosphorylate RB. This phosphorylation causes RB to release E2F, and E2F is then free to go and activate the genes that drive the cell into S phase. This is a crucial switch that controls the entry into replication. Moving along, the next major hurdle is the G2 checkpoint. A lot has happened between the G1 and G2 checkpoints. The cell has grown significantly, and most importantly, it has replicated all of its DNA. The G2 checkpoint is the final quality check before the cell enters the dramatic and complex process of mitosis. At this point, the cell is looking for two main things. First, is all of the DNA replicated? You can't enter mitosis with a half-replicated genome. That would be a disaster for the daughter cells. Second, is there any DNA damage? If a replication error occurred or some external factor damaged the DNA during S or G2 phase, this checkpoint will detect it. To manage this, the cell uses a class of proteins called CDK inhibitors, or CKIs. These are proteins that, as their name implies, inhibit the activity of cyclin CDK complexes. If DNA damage is detected, a signal cascade leads to the activation of these CKIs. They then bind to and inactivate the cyclin CDK complexes that would otherwise promote entry into mitosis. This gives the cell the necessary time to either repair the damage or, in severe cases, trigger apoptosis. The final checkpoint we'll discuss is arguably the most visually compelling. It occurs during metaphase of mitosis, where all the duplicated chromosomes are lined up perfectly at the metaphase plate, ready to be pulled apart. The M checkpoint is a meticulous inspector. Its sole purpose is to ensure that every single chromosome is properly attached to the mitotic spindle microtubules from both poles of the cell. Why is this so critical? Because if even one chromosome isn't properly attached, one of the resulting daughter cells could end up with a missing chromosome and the other with an extra. This condition, known as aneuploidy, is a hallmark of many cancers and often leads to cell death or severe developmental issues. The checkpoint works by monitoring the kinetochores, which are the protein structures on the chromosomes where the spindle fibers attach. If any kinetochore is not under tension, meaning it's not properly attached, the checkpoint sends a stop signal. This signal inhibits a key enzyme known as the anaphase promoting complex, or APC/C. Once all chromosomes are properly attached, the stop signal is removed and the APC/C becomes active. The APC/C then tags proteins like securin for destruction. Securin normally holds sister chromatids together. Once it's degraded, the sister chromatids are free to separate, and the cell can progress into anaphase. This is an elegant example of how a very small, precise molecular signal has a massive consequence for the entire cell. So far, we've focused on how proteins are synthesized and activated to push the cell cycle forward. But an equally important and often overlooked part of the process is the targeted destruction of these very proteins. The cell cycle is not a reversible process. Once a cell moves from one phase to the next, it needs to ensure it can't go back. This is achieved through the systematic destruction of key regulatory proteins. The cell uses a mechanism known as the ubiquitin proteasome system. Ubiquitin is a small protein that acts like a death tag. E3 ubiquitin ligases, like the APC/C we just talked about, attach ubiquitin to a target protein. Once a protein is tagged with multiple ubiquitins, it is recognized and degraded by a large molecular machine called the proteasome. This targeted degradation is what ensures the one-way street nature of the cell cycle. For example, the M cyclins that are required to enter and progress through mitosis are targeted for destruction by APC/C at the end of mitosis. Once they are destroyed, the cell can exit mitosis and enter the next G1 phase. Without this rapid destruction, the cell would be stuck in a never-ending cycle of division, which, again, is a defining characteristic of cancer cells. So, to summarize what we've covered today, I want you to remember these core concepts. The cell cycle is not a simple linear process. It's a precisely controlled sequence of events. The central drivers of this control are the cyclins and cyclin-dependent kinases. Remember, the CDK is the engine and the cyclin is the key. Together, they form a functional complex that phosphorylates target proteins. Checkpoints are the critical stop-and-go signals or guardians that ensure the integrity of the process. They check for adequate growth, proper DNA replication, and correct chromosome attachment. And finally, the cell cycle is regulated not just by turning things on, but also by turning things off. The targeted destruction of key proteins, especially the cyclins, is just as crucial for proper progression as their synthesis. Understanding these molecular gears and brakes is essential for grasping how a healthy cell divides and, more importantly, how a malfunctioning cell can go awry, leading to disease. That's it for today. Subscribe for more lectures. Thank you.